I just realized I wasn't recording on the YouTube. So we're 11 slides in and I just hit record. Sorry, everyone. Review on your own. Okay. So Punnett squares. Punnett squares are what that little thing was that I just drew right there that allowed me to understand how things are crossed. Punnett squares are pretty cool because if I said a person with freckles and a person without freckles, they had a child together, does that person have freckles? That's hard to comprehend for me, right? I have to look at it. This is a visual learning grid that describes the cross or the diagram of the individuals by using the possible parental gametes. This allows someone to determine the probability of what type of offspring are possible at that particular genotype and phenotype, okay? Some misconceptions about Punnett squares, for example, with this one, so this is a Punnett square. A misconception would be that, oh, the first offspring will have freckles, the second offspring will not have freckles, the third offspring will have freckles, and the fourth will not. Or if I have one of the offspring with freckles and the next one won't. That's not necessarily the case. This is showing the probability every single time that there is a possibility of an offspring, okay? Every time there's an offspring, there's a 50% chance of freckles. You could have all the siblings have freckles, even though there's a 50% chance that they didn't. It's also the reason why there's not a pattern to biological gender when a child is born. It's not like if they say, okay, if you first have a girl, the next one's going to be a boy. No, that's not the case. Some people have like 5 girls or 5 boys, right? There's no pattern to this. It's just saying every time you have a kid, you have a 50% chance of each gender. Every time you have a kid, in this case, you have a 50% chance of having freckles. And it's on every location on the chromosome. So these Punnett squares help us to determine what's the possible genotype that the offspring could have in this location. What's possible? I want to know my options, right? So here we have an example of freckles again. Your book does a lot about freckles. You could insert any trait here. You could say brown hair. You could say short toes. I don't even know if that's a real thing. You could insert any trait here, but let's just keep talking about freckles because it makes sense, okay? The parents at the top, they have freckles. We know that they have freckles because, first of all, their box is colored in red, and over here in the key it says that if the box is red, you have freckles. That helps, right? And maybe that makes sense to you. We also know that because they have a big F, small f, big F, small f, and if you have a big F, it means you have freckles. We know that both of those parents have freckles, okay? These parents cross, and they have an offspring, and these are the options to which that offspring could have freckles or not, okay? So you put each of the alleles on the outside of the square. So if I were starting this problem myself, I would start by drawing a square with a square on the inside, whatever that means to you. I like to do it like this and then draw a line here and a line here. Okay, we've got big F and small f, big F and small f. Okay, this particular trait does not pertain to sex at all, like this gender of this. But what does matter to some people, and not necessarily to me, is some people like to keep the male and the female on the same side, but it doesn't matter necessarily because you get the same answers. But for you, if you're somebody who's like, I'm type A, I need to stay organized, I need to do the same thing every single time, put the female or the eggs on the top and the sperm on the side, okay? You don't have to, but you can. All right, for this example, like I said, it doesn't matter. So here, what you do then is you go to each intersection of the square. So this one goes F, F. I went here and I went here and I got this. Does everybody see where I got that? Yes, okay. try the other two without looking at me if you want to on a scratch piece of paper. I go here and here. I get big F, small f, big F, small f, small f, small f. Is that what they got here? Yes. Same thing. Big F, big F, big F, small f, big F, small f, small f, small f. Okay? So here's a couple of things. The first word on here that we haven't talked about yet is a phenotypic ratio. You may have maybe heard of a ratio before in a math class or a stats class, but we could do a ratio of color of shoes, right? You could have black colored shoes or other type of shoes. And we could have like a 6-to-3, or let's do a different number, a 5-to-3 ratio of people having black shoes to a different colored shoe. Okay, that's a ratio. Here we're interested in the ratio to know either the trait or not the trait. Phenotypic ratio, meaning what's the ratio of people who have freckles to not having freckles? Well, in this case, we can see that 3 out of the 4 boxes are red and 1 out of the 4 boxes is white. So 3 to 1 are freckles to no freckles. Freckles to no freckles, okay? The phenotypic ratio is different than the genotypic ratio. So let's add this over here, okay? To have a genotypic ratio, this is about the different alleles. So how many of them are big F, big F? How many of them up there have 2 big F's? 1. How many of them have big F, small f? 2. How many of them are small f, small f? 1. That's a genotypic ratio. Okay, a phenotypic ratio is showing how many of them have freckles. How many? How many of the individuals have freckles? 3, yeah, thank you. And then how many have no freckles? 1. All right. You guys see the difference in those two numbers? So this one and this one are telling us the same things, but they're asking for different things, okay? The genotypic ratio is saying how many of each of these letters are present. What are the possibilities of having a big F, small f? Well, the probability is you'll most likely have a big F, small f because that's the most frequent one up there. What about freckles to no freckles? This one is showing you just how many have freckles, or what's the option of having freckles. The option of having freckles is 3 to 1, so you're most likely going to have freckles if this is your parents. Okay, that's what's showing the difference here. Remember, phenotype, the physical trait. Genotype, the gene code. Okay. All right, practicing with Punnett squares. This is the same thing that we just went over. Would a Punnett square involving a man with a genotype of FF and a woman with FF look like? We just did that. It's the exact example on the last slide. Okay. Now it says to practice the ratio, which we just did. Genotypic ratio is the number of offspring with the same genotype, meaning how many offspring, or what are the possibilities of the offspring having a big F, big F? The probability would be 1 to 2 to 1. The phenotypic ratio is the number of offspring that have the outward appearance or physical trait. Or what is the word that is describing what's going on? That would be the phenotypic ratio. That would mean that it's a 3 to 1 ratio of this person having freckles. Okay, so we wrote that up here on the PowerPoint, even though we did it on the board as well. Is anybody lost? Does anybody need me to do this again? Okay, that's good. okay? This is called a monohybrid cross. This is where we cross parents are identically heterozygous at one gene pair, okay? So an example would be on the left would be a monohybrid cross, an experimental cross in which the parents are identically heterozygous at one gene pair, okay? So, for example, on the left, they have the same genotype for all of their offspring. And B, they have it where their offspring are similar in two different ways, okay? This is determining if a dominant phenotype is heterozygous or homozygous. This slide doesn't really make sense to me, so we're going to keep moving on. Because a monohybrid cross is just saying that you're crossing one thing. So moving on. Possible gametes for two traits, okay? This is talking about meiosis. Maybe you've heard of meiosis and mitosis. We do talk about it in a different chapter, but just a brief review if you don't remember, okay? Can you guys still see if I'm right over here? I guess I could go over there. Okay, meiosis versus mitosis. Okay, this is also a future grade exam question because it involves lots of different chapters. Okay, meiosis is your sex cells. So this is your sperm and egg cells. It has two divisions. Okay, so that it has half the number of chromosomes than what it starts with. Okay, so although it's hard to process in your head, for males, this is constantly happening. Meiosis is the production of sperm cells. Okay, so what's created are sperm cells at the end of this process. For females, females are born with all the eggs that they will have throughout their life. While they do mature at puberty and throughout their life until menopause, the production of egg cells happens at essentially growth and development. Okay? Mitosis, these are all the other cells except for your sex cells. So I'm just going to put the rest. Okay, these are the rest of the cells. This is your skin cells that turn over every 2 weeks. This is your liver cells. This is your insert type of cell here. Okay, this only has one division and it creates two identical daughter cells, which we're thankful for, right? When I get new skin cells, I'm really glad that it has the same amount of chromosomes as the skin cell that came before it. Right? That's pretty cool. Same thing with that. So meiosis versus mitosis. The difference here, you can see that this one has two divisions, this one has one division. This one has 4 daughter cells. Okay, so we will talk about that in the coming chapters, but let's talk about it here with genetic inheritance, okay? So we have mitosis 1 and mitosis 2. There's your two divisions, right? Meiosis 1, we divide again, okay? Here is, for example, an egg cell, okay? We want to produce more egg cells from this egg cell. So we divide and divide again. Now, all of these, we have 1, 2, 3, 4, 5, 6, 7, 8, 8 possibilities that come from this egg cell. And notice how now they have half the number of chromosomes than what we started with. So first thing we do here is we have 4. 1, 2, 3, 4. We have a big blue, a big red, a small red, and a small blue. Those are called chromosomes. Let's just worry about their size and their color right now, okay? First, let's only worry about the left side. First here, we have the option where they reproduce, they replicate. So now we have, you see that it looks like a little X? We have a big blue X, a big red X, small red X, small blue X. Okay? We just replicated. All of these got a little buddy called a sister chromatid. Okay, they replicated. They replicated because they know we're going to divide. We want to make another cell. We want it to have half the number of cells that or chromosomes that we started with. So we replicated, now we divide. Now do you see the small x's here, the big or small blue X? So small colors separated and the big colors separated. We then divide again. So now we have one that has a small red, big blue, big blue. Small blue, big red, small blue, big red. Okay, those are how we've decided to divide. That's one option. Okay, but what if they divided in this other way? So they did the same step here. These are the same steps. And then we divided, but they decided to divide by color this time. Ooh, that's fun. Right, so but now we have a small red with a big red, small red, big red, small blue, small blue, big blue. Both of them went through meiosis. We came up with different options here because of how they divided. Okay? This is called independent assortment or crossing over. These are cool reasons why you may not look like your sibling if you have a sibling, a brother, a sister. It's also gets a little complicated with twins, which we don't worry about as much. But just know that just because there's an egg cell here and it divides, it doesn't mean that we're going to get the same outcome every time. And then you have another individual, if this was an egg cell, so then another individual is making a sperm cell that also is going through this very similar process, and then the randomization of which sperm fertilizes with which egg. And another thing about this slide is this is two different processes. That's why it says either or. This is not all one, the same process. So replicate, divide, divide. Replicate, divide, divide. Those are two options. Okay. A dihybrid cross is the opposite of a monohybrid cross. This is the cross of two trait cross. For example, we know that some traits are linked together. For example, this one, it just says big F, or big S, in the top right-hand corner here, right here. You guys can kind of see that. It's hard to see right here. This is also figure 21.6 in your book. It's just showing how at this specific location, they have the option to pass on big F and big F and big S, and this person is passing on small f and small s. And so when they cross here, the individual ends up being big F, small f, big S, small s. Okay? When you cross somebody who's heterozygous, the results are always 9:3:3:1 for the phenotype or the genotypic ratio. Okay, so let's talk a little bit more about that. Essentially what it's saying is, so this is a Punnett square. We're just taking it up a notch and getting more options. So essentially you can think about it as this is a Punnett square, this is a Punnett square, this is, and this is, okay? But at every location, they have two letters. And those two letters are linked together because it's a dihybrid cross. So this first individual is big F, big F, big S, big S, okay? So here, that individual is only going to pass on a big F and a small s, okay? So in every location, big F, big S, hard to say 10 times fast, big F, big S. Okay, the other combination we can have is big F, small s. We can do that twice. And those are our options here. Then you cross at every intersection. So here we have big F, big S, big S. Okay, and at each intersection they have an option here. Okay. This is maybe freckles are tied to lighter skin tones. It's not always the case, but it could be. And that's why things get a little tricky with genetics is because it's how things cross, how things divide, and the randomization of the sperm and the egg cell. Here's just a little bit more zoomed in look at one of the columns, so you can see that a little bit better. Remember, a 2-trait cross just takes into account 2 traits that are linked together. It makes it even more random for something to happen, okay? So this is just the phenotypic ratios of common crosses. So if you're somebody who likes to recognize patterns and skip ahead a couple of steps because the Punnett squares, you just don't have time for that, you could go to this table and recognize patterns. So when a monoheterozygous and a monoheterozygous go together, it's always going to be a 3-to-1 dominant ratio, if you like to recognize patterns. If a monohybrid heterozygous and a recessive homozygous come together, it's always a 1-to-1 ratio for dominant to recessive. Dihybrid is where we have 2 traits. If you have a dihybrid heterozygous, it's always going to be a 9-to-3 to 3-to-1. And then a dihybrid heterozygous to a recessive homozygous, it's always going to be a 1-to-1 to 1-to-1. If you like to recognize patterns, you do not need to memorize this table by any means because you could work it out. You could figure it out through Punnett squares. Okay. So this little nifty diagram is what we like to call a pedigree or a family tree, okay? And this shows relationships through different generations. So you could essentially figure out a pedigree of your family with a specific trait, okay? For example, with this trait, we've got different generations. We've got generation 1, 2, 3, and 4. The other way to look at this is like maybe these are the great-grandparents, and these great-grandparents had these kids. These kids had offspring with these kids, and then these relatives. Wow, that was really fast. These are cousins, right? They just had offspring together. That's weird. So the way that you read this is when there's like a T intersection, you see where there's like a line between them and down. That means that they had offspring together. What's kind of interesting to think about with this is that marriage doesn't matter, right? This is not a sociological table. This is a biological table. We want to know if it's a male and a female and if they had offspring. That's all we really care here, and if they have a trait or not. So the way that you know this is that the biological females are going to be circles, biological males are going to be squares. Okay, so in every intersection, male, female, female, male, 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 female, 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 male, female. Okay. The next thing we can tell is that whatever trait we're looking at in this key, it says if it's highlighted yellow, they're affected by whatever that is. Say it's Huntington's disease. Do they have a specific example here? Huntington's disease is a recessive disorder. So if somebody has Huntington's disease, they're going to be highlighted in yellow on this pedigree. Okay? If they are capital A, small a, they're a carrier of that trait, but they're unaffected. That means they walk around without the disease, but it's possible for them to pass on the trait. As you can see, essentially the great-grandparents, I guess you could say, of these people, the female had the trait, but it skipped a couple of generations, and then they finally started to see it in a couple of generations here. Okay, so essentially these people had offspring here. These 2 people had 2 females. They did not have these. These are random other people. I guess not random in a sense, but you know what I mean. They could come from different pedigrees. This just means that they had offspring. This offspring here, and these offspring here. So essentially these are sisters that had offspring, and their offspring had offspring, so their cousins had offspring. In this example, that's kind of weird. What you'll see here is that we don't know the second allele sometimes of that person. When I'm going through a problem like this, I usually leave it like A blank, meaning I know it's a dominant trait, but I just don't know what that second allele is, and I can't make an assumption because it could throw off the whole table. Okay? But here, big A blank, or big A blank, big A small a, okay? Big A small a. You can still go through all the possibilities of that offspring, and because of this pedigree, we can see that they're unaffected because it's shaded in white. So we know that that person has to be any of these, but not that one. Possibly not that one. It's possible they could be this one if this is a big A. Okay, that's just a possibility, but we don't know that for sure. So leaving that blank like that shows that you know it's a dominant trait, but we just don't know that second allele. And then when these relatives came together, they don't have whatever this trait is, say it's Huntington's disease, but they had offspring that did. Do you see why? They both have a small a here, and when you cross them together, you can have the possibility of that. If you can't see that right away, when in doubt, draw it out. Draw that Punnett square. Draw an A with a small a, an A with a small a, just like those are crossing. And then draw the options. We'll be able to see that the two small a's can come out as a possibility. So this is showing a couple of different things for us. This is the first picture we've seen of a pedigree. This pedigree shows the relationship between generations, which are the different rows. We can see who biologically had offspring. Remember, it's just the science, right? We want to know if they had offspring or not. We don't care if they got married and divorced 6 times. If they had offspring, they're going to be on this table, right? If they don't have offspring, they're not going to be on this table. Okay. Here's another example. We don't have any cousins doing their thing anymore in this example, so that's nice. Individuals that are homozygous dominant and heterozygous will have the disorder. So we have a pedigree here. Remember, the circles are females, the squares are males. All right. And we can see the relationship to that inheritance. What I'd like us to also note on this last slide, it says autosomal recessive disorder at the top of the slide, okay? The word autosomal just means all of the cells except for the sex cells or the sex chromosomes. So this is also, we could call this autosomal. Autosomal. Okay. So what's important to know about this is that there's 4 different types of ways that things can be passed down genetically, okay, at a certain trait. Just because having freckles is dominant doesn't mean that you always have the dominant trait at every single allele. You could have freckles and then be recessive for cystic fibrosis and have cystic fibrosis and freckles. It's possible. There are different locations, okay? So the ways that things can be passed down genetically, we'll just go over here. Write it down. Okay, you can have one we call autosomal, and then we have and the other we call sex-linked or X-linked. Okay, and at each of these, it can either be passed down dominant or recessive. And same here. These are 4 patterns of inheritance. And they tell you a lot about something. So they could say, if color blindness is sex-linked recessive and the individuals look like this, is it possible for their offspring to have color blindness? Okay, so that's how things can be kind of told to you. So the next one, that first example here, we showed autosomal recessive, meaning that it's normal traits, gender doesn't matter here, or the sex chromosomes don't matter here, just whether or not they have freckles. And it's passed down recessively. This one, meaning that you have it if you have the dominant trait for it. So this one, you can see that all the ones that are affected have a big letter A in their diagram, okay? We can see how that is passed down. If you have two small a's in this table, you don't have it. Okay, this is just a different location. This could be for brown hair. It could be for Parkinson's disease, whatever that is. It's just a different trait. Okay, there's thousands and thousands of different traits that communicate together to make you who you are. Okay, the next one we have is just talking about autosomal recessive disorders that may interest you or that you maybe have heard of before. The one I mentioned before was cystic fibrosis. That one seems to be somewhat common. I say somewhat because most of the time people know what that is. What's happening is the chloride ions do not pass normally through that cell membrane. It's causing them to have this mucus in the lungs. That's why if you've seen somebody who has cystic fibrosis, they kind of have a lot of maybe coughing. It's because they've got mucus in their lungs, okay? This is a genetic disorder and it's passed down autosomal recessive. What that means is that you could have two unaffected parents that have a child that has cystic fibrosis. Well, what does that mean? So when we go through these steps to figure out what I'm talking about here, the first thing you should ask yourself is, what does it look like to have that disorder? What does it look like to have it? Well, in this case, if you have an autosomal dominant, you look like big A, or big A, small a. If you have an autosomal recessive, which is what we're talking about here, if you have it, you look like small b, small b. Okay, we'll worry about X-linked or sex-linked in a second. So the example of cystic fibrosis would be if you have two parents that do not have cystic fibrosis. Right, both of these parents don't have cystic fibrosis. Hopefully you can see my handwriting back there. And we have the possibility of having a child that doesn't, but they also have the possibility of having a child that does. And that is how something like that is passed on. Autosomal recessive. Let's see, we're doing a bit on time. Okay. Also, the other one is Tay-Sachs disease. This is just what there's a lack of enzymes that break down the fatty acid proteins in lysosomes that results in accumulation. This just allows you to not fight off infections as well if you have Tay-Sachs disease. Here's a couple other autosomal recessive disorders. Autosomal meaning not your sex chromosomes. Recessive meaning that it's the opposite of dominant. PKU is the lack of an enzyme needed to make a certain amino acid. It affects the nervous system, so this can cause some like trembling, twitching, it's as if your body's reacting to stimuli that's not there. Sickle cell disease, we talked about this with red blood cells. This is also an autosomal recessive disorder. We talked about this, right? If you have two individuals, like I can use this table again. If you have two individuals that are resistant to malaria, because if you look like this, you're resistant to malaria, and they cross, they can have a possibility of passing down sickle cell disease to their offspring. It results in like a raisin kind of sickled shape to their red blood cells, which they can't effectively transport oxygen to the rest of their body, causes dizzy, fainting, even death at some points if it gets out of hand. Kind of serious. Here's an example of Tay-Sachs disease. We don't really talk much about Tay-Sachs disease, so I'm just going to keep moving on. Just know it's one of the disorders. So now moving on, we have autosomal dominant disorders. So this is another pattern of inheritance. Remember, we have autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive. This is the opposite of autosomal recessive. Some of these are Huntington's disease. If you have Huntington's disease, the Huntington protein has too many glutamine amino acids, and it leads to progressive degeneration of brain cells. So if you can guess what that means, if you're kind of progressively degenerating or kind of breaking down your brain cells, you probably are going to have some loss of function over time, right? Osteogenesis imperfecta, this is a defect in collagen synthesis, which remember collagen makes up your hair, skin, and nails, the elasticity of your skin. It results in weakened brittle bones, which makes sense. If we're not producing collagen as well, then we're going to have brittle bones. Brittle bones, easily broken, can't really hold yourself up very well. Marfan syndrome, this is a defect in the production of the elastic connective tissue in the protein fibrillin. It results in the dislocated lens, long limbs, fingers, caved in chest, and a weak wall of the aorta. So essentially what's happening here is the elastic connective tissue is just not working as well. So a person with Marfan syndrome is going to have really long fingers. They're going to have a caved in chest, and they're going to have a weakened wall of the aorta. We know that the blood from the aorta goes to the rest of the body, right? We just took a test over that. So if you have a weakened wall of the aorta, you're just not getting blood to your extremities as well. It makes physical activity, even just like general walking and things like that, very difficult. Okay, here's a little bit more about Marfan syndrome. It attacks your skeleton, your heart, and your blood vessels, your eyes, your lungs, your skin. It does a lot of different things to your body. Okay, so the skeleton, right? Because the elastic connective tissue and the proteins aren't working as well, your chest wall kind of starts to deform. So think of like an inverted chest wall. You have thin, long fingers and arms. You may have scoliosis or a curvature in your spine. Flat feet, long, narrow face, and a loose joints. Okay. You could also see heart and blood vessels. You're going to have a mitral valve prolapse. So we know where the mitral valve is, right, in the heart, on the left, anatomical left side of the heart. You can have an enlargement of the aorta, which also makes it really weak. So you could have an aneurysm. The eyes, your lens of your eyes can be dislocated. It can cause severe nearsightedness. I don't know about you, but I always get it mixed up. If you are nearsightedness, it means that you can see near, up close. So I'm nearsightedness. So if I were to take out my contacts, I couldn't see any of you. So that's what nearsightedness means. Lungs, you'll see a collapsed lung, which makes sense if we're having a curvature of the spine and a caved in chest wall. It's probably going to result in a collapsed lung. And then the skin, you're going to see stretch marks in the skin. This is because of the stretching of the membrane that holds the spinal fluid. So you're stretching your back and you're in different ways and things like that. This is Marfan syndrome. It's an autosomal dominant disorder. It's passed on. If you have Marfan syndrome, then you have the dominant trait. A little bit about Huntington's disease and cystic fibrosis. So on the left, Huntington's disease, remember, it degenerates your brain cells. So on the top, you can see that there are many brain cells of a normal brain. And on the bottom, you can see that there's just a loss of brain cells, okay? Loss of brain cells can lead to a lot of negative side effects, such as you aren't able to respond to stimuli as fast, right? Which is probably not great if you're trying to get up and walk. It may take you a little bit longer for your brain to physically tell your muscles in your body to get up and walk. Then you start to lose fine motor skills, like picking things up like your pen, talking, eating, things that we all, sometimes take for granted every single day. Cystic fibrosis, this is just where there is a clogged chloride channel and there's a mucus in the lungs. So what this person is wearing on the right is a percussion vest. So if anybody's ever been in percussion before and band, right? All the people that make the pounding noises, that's essentially what this is doing to the chest. It's kind of like a vibrating chest piece, and it's going to try and break up that mucus so that the body can break it down and not have like plugged up in the lungs. We've come a long way with cystic fibrosis. I had someone in my high school who passed away at a young age from cystic fibrosis, but nowadays, if there's a child that's diagnosed with cystic fibrosis, it's not necessarily the case because of the advancements in medicine. Okay, so this is talking about pre-implantation of genetic diagnosis. So knowing whether or not parents have a certain trait may trigger them to get a genetic test or see if they have a genetic disorder either before or during while they're having an offspring. So if the prospective parents carry an allele for a genetic disorder, So maybe you know that you're a carrier for cystic fibrosis. They may seek assurance that their offspring will be free of this disorder. They can do that through in vitro fertilization, or where the zygote divides. When the embryo has 8 cells, one may be removed for genetic testing. So this is very, very early on in IVF, okay? Also, I'm just stating some facts here. You can have your own opinions about all of this, completely fine. This is when only the embryos that will not have the genetic disorders of interest are placed in the uterus to continue developing. So essentially what this is saying is pre-implantation. So before the zygote is implanted into the carrier, or essentially the female carrier, most likely like the mother, not always the case in like surrogacy, but before that, in a lab, in vitro, they fertilize a sperm and an egg cell. And through mitosis, those cells started to grow and divide. When they got to 8 individual cells, think about how small that is. They took one of those cells out for genetic testing, and then they will remove that cell and leave the rest in there for it to continue to develop in the uterus. A lot of science here, right? This kind of blows my mind that we're able to do this. But as you can see at the top left, there's an 8-cell embryo. An embryonic cell is removed. The cell is genetically healthy, so they put it back, and the embryo develops in the uterus normally, and then I guess it goes right from, 8 cells to a baby, I guess. And then here's an example of where the woman is a heterozygous. They get the egg out. It's a polar body. It has a genetic defect, so they recognize that one of the cells has a genetic defect. And so what they do is then they take it out and they put in a nice, like a better cell, and then the embryo develops normally in the uterus. So this is just called pre-implantation genetic diagnosis. Okay. So we have something called a polygenetic trait or polygenetic inheritance, meaning that it's 2 or more sets of alleles that govern one trait. Okay, so each dominant allele codes for a product, so these effects are additive. For example, this is like height or skin color or things like that, where having more of something results in a different trait. Okay, so polygenetic, poly meaning many, okay? These result in a continuous variation of phenotypes, and we'll show a cool graph here in a couple of slides. The environmental effects can also cause intervening with the phenotypes. Skin color ranges from very dark to very light, okay? You can also think of this kind of globally, right? Darker skin colors in the past have come from people who are closer to the equator, having ancestry of closer to the equator, a lot more sun exposure over time, okay? Now passed on through genetics. Somebody with a darker skin color does not necessarily have to be from somewhere near the equator, right? It's just a lot of very interesting things to think about. This is also height. It varies among individuals and humans, right? We would all take our heights in this class. We'd all be a different height most likely, or somebody would be the same height. It's possible, but it's varied. If we were to take the heights of everybody at Wartburg College, you'd also see a variety of heights, okay? There's also studies that have been done that have shown that humans are taller than they were 100 years ago. Why that is, I guess I don't know. Probably through crossing and dividing. It's also a multifactorial trait, which means that it can be influenced by the environment, which is like I was talking about. Think about the nutrition that we have now that maybe people didn't have 100, 200, 300 years ago. We're really lucky in a sense here at Wartburg College. You guys have the access to food pretty much at any time, right? Any time of the day. Is there anything open 24/7 around here? Pretty close. Late night snacks?
And is open till 11.
Yeah, see, 11 o'clock. If you were a cave person back in the day, you'd have to go out and get your hunt and gather for that food, right? Go eat your berries off the plant, all right? think about the nutrition in the food that we have access to all the time now, right? If you're hungry, you can just go drive to McDonald's, whatever that is, okay? Skin color is also influenced by sun exposure, okay? But these also are a multifactorial trait, meaning there's a lot that goes into it, a lot of different facts or different things like the environment and genetics, but also more of it, so more nutrition. Doesn't mean that if I go eat 10 cheeseburgers, I'm going to be 7 foot tall. It just means that over time, because there's been a bigger supply of nutrition and more access to food, people are getting taller and I guess also wider in a sense, right? Same thing with skin color. So here's a really cool diagram. I think it's pretty cool. It's a way to think of this polygenetic inheritance, okay? The top one there is an example of skin color. Now, it doesn't mean that every skin color falls into this graph by any means. It just means that the very, very almost albino pale skin compared to very dark complexions, and then you've got kind of like this middle range where it's a little bit less frequent to have these colors of skin, and then as you get closer into the middle, it's more possible to have that. It's a number of dominant alleles that you have in that specific trait. It ends up being a bell-shaped curve. Same thing goes with height, right? You have height. This is just in inches in this example here, but being really tall and being really short is just kind of rare, right? It just is not as common. And that's okay. It just means that most people nowadays in the height range kind of fall into that middle category. It doesn't mean that there was anything done to do that. It just means that with the data that we have, it kind of falls into a bell-shaped curve, okay? These are polygenetic inheritance. So if the exam question would say, give me an example of polygenetic inheritance, there's two, right? You can talk about how it's kind of like a spectrum almost of different skin colors or different heights. You could even do like shoe size or things like that, okay? That's pretty interesting, I think. Okay, this is an example of how environmental things influence phenotype, okay? So we have this Himalayan rabbit, all right? And the coat color of this Himalayan rabbit was influenced by temperature, okay? There is an allele responsible for melanin and that produced the appearance to be active only at lower temperatures, okay? So when it was lower temperatures, this allele for melanin would react and it would have like a little bit darker pigmentation. The extremities have a lower temperature and thus the ears, nose, paws, and tail are dark in color. So this is just showing how this rabbit, its traits are reacting to the environment and it's creating where it had lower temperatures. Think about it. Why do we put a hat on our head? Most of the heat escapes to the top of our head. Same theory, you got a lower temperature. That's why those ears are darker in color. You're touching the snow on the bottom of the floor. That's why it's a little bit darker in the paws there. Just an example of how environment can affect phenotype, right? You could think of it as food. You could think about it as sun exposure, things like that. Think about flamingos. Flamingos are actually born a grayish, whitish color. And then after eating crustaceans and shrimp and things like that, they turn that pretty pink that you think of when you think of a flamingo, okay? It's how your environment influences your phenotype. But I guess you are what you eat. Okay. Incomplete dominance. So this is a type of genetic inheritance, incomplete dominance, meaning that both traits are just sort of present. So if you have parents, one has curly hair and one has straight hair, it's possible for the offspring to have wavy hair, where it's like kind of in between, kind of on a spectrum, right? If there's no like exact right answer to that. Incomplete dominance. Something that's called co-dominance would be where they're both present, would be like blood type, and we're going to talk about that in a second. But incomplete dominance, I like this example because it kind of makes sense. If you have a curly hair individual, a straight hair individual, and they cross and have offspring together, it's possible that that person has like a wavy hair type, Okay, that's what incomplete dominance means. We're not going to talk about that slide. Codominance is the example like blood type, where both can be present at the exact same time, like your coworker, your co-captain, okay? This is where if an individual who has type A blood and an individual who has type B blood come together and have an offspring that has AB blood. It's not like the wavy hair example where you'd like sort of have A and sort of have B. That's not this. This is codominance, where they're equally present. We also have multiple allele inheritance. This is a gene that exists in several allelic forms. This is a person who only has 2 of the possible alleles. A good example is the ABO blood system, where A and B are codominant, but then we also have an O. Okay, so multiple allele inheritance, just meaning that these examples, we only had F's to take from, right? Codominant, saying we have A and B. Multiple allele inheritance, we have ABO, right? ABO blood type, because you could be A, you could be B, you could be AB, or you could be O. Multiple allele inheritance. The O is recessive to both A and B. Therefore, you have this blood type, you must have 2 recessive alleles. This is also an example, I'll use chalk. Okay, so if you have A, B, AB, or O blood types, your genotype, this would be your phenotype, by the way, because that's what type of blood you actually have. Okay, if you have a genotype, you have a couple of possibilities here. If you were type A blood, you either look like AA or AO. Okay. If you're type B, you're either type BB or you're BO. If you're type AB, you're just AB. There's no other options. If you're O, you're just OO. Okay? This is also how it's possible for a person with type A and a person with type B to have offspring that's O. Wait a second, let's see that, right? So if you have an individual that's type A and an individual who's type B, and they cross to each other, okay, it's possible that if their second allele is an O, you could have somebody who's OO. This is called multiple allele inheritance. So blood type takes into account a couple of things. Notice how here I didn't put AO, BO. When an A and a B come together, they're codominant. A and O come together, it's just dominant recessive because O is recessive. This is codominant. Bless you. This is codominant. And so is this one. Sort of. Okay. Here would be dominant recessive, dominant recessive. So this person has type A blood, this person has type B blood, this person has O blood. That's what multiple allele inheritance means. Okay, we just kind of wrote that on the board. All right, possible genotypes and what their phenotypes would mean. Remember, genotypes are the gene code. Phenotype is the physical trait. Here's another example. So the book uses I's, like the letter I. Essentially what I do that I think is easier is just to eliminate that I and just write where there's a little I, just put O. So essentially what they did here was exactly what I showed. A-O-B-O. But if the I's make more sense to you, go ahead and do it, okay? The key here is just showing that there's different types of phenotypes that are possible. The phenotypes that are possible are A, B, and O. So if these 2 individuals were to cross and have offspring, they have the possibility to pass on any of the blood types to their offspring. The phenotypic ratio is 1 to 1 to 1 to 1 because we have all of the possibilities here. Okay, So now we're moving on to sex-linked inheritance. Those are the two over there that I haven't talked about yet. So the 4 patterns of inheritance are autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive. Okay? We also have these funky ones, like we said, multi-factual, for like a poly-inherited trait. We have codominance, we have multiple allele dominance. And now we have sex-linked inheritance. This is where the traits are passed on the sex chromosomes. So you've maybe heard about sex chromosomes before. Biological females have XX, biological males have XY. That's just the chromosomes, okay? And it's saying that whatever these traits are passed on that chromosome. It's also why color blindness is more common in males. And we'll talk about that here in a second. We have the options here to have an X-linked trait and a Y-linked trait. We don't really use Y-linked traits in this class. So when you see the word sex-linked traits, just think X-linked traits. Okay, so here's that color blindness example. Now, something to think about is just how a female and a male come together to make offspring, okay? So here, the female is always going to give an X chromosome. All right, let's think about that for a second. Remember, we can only give one allele at a time, okay? So no matter what, the female up here, male here, the female is always going to give an X chromosome here. Because male or female, we both have X's. The male either gives another X and it creates a female, or it gives the Y chromosome and it creates a male. And that's why there's a 50% chance of having males and females, you see that over there, with every genetic cross for sex-linked trait, let alone just what's the gender of the infant or baby or whatever it is, okay? So now that we know how that is crossed, let's talk about X-linked inheritance for color blindness. If you have color blindness, it's going to be indicated by the light or the white colored square, being that you have to have it, it's a recessive trait. So now we know that color blindness is X-linked recessive. If you have sex-linked recessive trait, you look like this. You're either a male and have a small letter, or you're a female and have 2 small letters. Okay, if you have an X-linked dominant, you're either X big B Y, or X big B, X little b, or X big B, big B. Okay, those are how you draw those. Okay, so let's think about this one. We have an egg and a sperm. They're going to come together. We're going to see if any of their offspring are going to have color blindness. Okay. First of all, we notice that neither of the parents have color blindness. The male has a big B. The female has a big B. Neither of the people who start with have color blindness. Now you've got X big B that come together. You just kind of do the intersection. X big B, X big B, X big B, X little b, X big B Y, because here's the Y, X little b Y. So what happened was all males get their X chromosome from their mother every time. That's where they get their X chromosome from, because they get their Y from their father. So here we had an example where the mother was a carrier for color blindness and passed it on to her male children. There's no possible way that any of the females could have color blindness that were her offspring. It's not possible. It's possible for that one on the right to be a carrier, but it's not possible for them to have it. All right. All right. This is an X-linked disorder. This is also showing X-linked recessive. All right, I won't go through it too much just because you guys can read that on your own, and we kind of went through the example already, but just remember how to read a pedigree. Each row is a generation, okay? When there's a T intersection, meaning there's a horizontal and a vertical line, that means that they had offspring, Okay, and remember, it's just a biological table. Here are some different X-linked disorders. We already talked about color blindness. There's a type of muscular dystrophy, Fragile X syndrome, hemophilia. These are often found in males than females because recessive alleles are always expressed. So that means that because a male always gets their X chromosome from their mother, if the mother happened to pass on the recessive X chromosome, no matter what happens, that male is going to express it. But that doesn't mean that for females, because the female could inherit a dominant X from the father and a recessive from the mother, so there's less chance of a female having an X-linked recessive disorder. You guys can read through those. We've got, let's see here, just a couple of slides here about muscular dystrophy and just showing how there's abnormal tissue. It's more common in males, this type of muscular dystrophy anyways. And then the last one being hemophilia. This is a really big pedigree, but it's of the royal families, okay? And if you wanted to take a look here, this is kind of interesting. So you have Queen Victoria and Edward that have Queen Victoria. Queen Victoria then has offspring with Albert, and they have this one, 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 and this one, and that one. And some of those words you may remember are Alice, Louis the Sixth. I don't remember Roman, is that the fourth? Fourth, yeah. Helena, Beatrice, all these royal people. And then they have offspring. 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 Uh-oh. What do we have here? Little incest is the word.
First cousins.
What's that?
Those first cousins.
Yep, first cousins. So these are sisters who have offspring and their offspring have offspring. So that's interesting. It's also why in this particular, so the reason that this happens where you see cousins having offspring a lot of the time, where's another example of cousins? Oh wait, is that? Yeah, that.
No, I think,
No, that's
I
not
think,
so.
I think some of it is just how it's spaced out.
Yeah, just how
Like
it's
the one
spaced
on
out.
the left is just.
For sure cousins here, but the reason that in the royal families where they didn't have a lot of information about genetics and things like that, why they wanted to do this was to keep their names in the family, because having royal families was really important at that time. But anyways, it's also how some types of genetic disorders happened and how they, for example, can be passed on through different generations. So this is like a very interesting table if you want to work through it sometime, but yeah, there's a lot to look through. Just remembering that circles are female, squares are males. Cool. That's all I have for you today. You do have an assignment on my Wartburg under coursework. I'm very close to finishing your exams. I'll probably finish grading them today. If not, I'll be watching The Bachelor. There's a chapter 21 lecture assignment. There's also an extra credit assignment this exam. So with this extra credit assignment, it's over chapters 19 through 22. It's a review. It goes in your homework category. So when I grade your exam, this extra credit wouldn't go in that category, but if you've missed maybe 1 or 2 points on your homework, this is a way to get some points back from that, okay? My recommendation would be to do it towards the end and use it as a review, like right before the exam, because as you can see, it's over chapters 19 through 22, and we just went through chapters 21, so you may not know about 3 of those chapters yet. But, I mean, you can do it whenever you want. If you get 1 point on it, I'll give you 1 point of extra credit. I think it's worth 10 points, if I remember correctly. So, that is all I have for you. That will be due on Friday. We have no lab on Wednesday because you guys have a break. Woohoo! An observed college break.